Thomas Werner from Munich, Germany, who has an adjunct appointment with the University of Michigan here to give a special seminar. Thomas is a PhD who attained his PhD from the University of Munich in cell and molecular biology, because at that time there was no bioinformatics PhD available. He, however, established that field in the Helmholtz Institute, which is a similar system to the Harvard Hughes Institutes here, built the first bioinformatics group up there, and then in the 90s established the first bioinformatics startup company in Germany, was the founder, CEO, and CSO of that company until a few years back. And at that time, we actually connected during my time in Munich and started to learn the power of these approaches and tools and have deployed them eagerly since then. And we are very fortunate to have Thomas now regularly in Ann Arbor and helping us with our research projects. And he will give us an overview of his ongoing work, how to integrate genome-wide association studies with multiple lines of evidence to identify disease mechanisms. If you find some of these strategies interesting and compelling for you, Thomas is in town until, I think, Friday. So would be available for follow-up discussions either directly after the seminar or thereafter. Feel free to reach out to me and we can see if there's an opportunity for you to meet. And with this, and no further ado, Thomas, the stage is yours. We are looking forward to your presentation.
Thank you very much. So actually, I'm going to tell you a nice little story on an afternoon like this. No heavy science here. Just a nice little story that you hopefully will enjoy. And the point is, this is between Munich and Ann Arbor, as we already said. And together we're doing more research. And I hope to convince you in more than one sense that we do this. The basics, the cornerstones of everything I'm going to talk about are two things. More cassettes, which a few of you are aware of and most are not. And lines of evidence, actually multiple lines of evidence, which is the important part about that. But let's start out with what on earth are more cassettes? Well, the very first thing they're part of the genomic sequence, like a reading frame. You need a computer to read it, but it's nothing we made up. It's existing stuff from the genome. We just use it like you use a reading frame to understand what protein is encoded. And we use more cassettes to understand how an RNA is actually regulated. So a bit more in detail, it's kind of the programming language of promoters and enhancers to some extent. And it's based and it's written with transcription factor binding site, with EFBS here. And little sequences that have the ability to bind specialized proteins. They are notoriously wobbly. So you have to describe them by weight matrices that don't have a clear-cut consensus. And they're also a bit promiscuous in terms of what they bind. So transcription factors and their binding sites have an N-to-M relationship, not a one-to-one, which is important. So I call them multiple organized regulatory elements, cassette. Why cassettes? Because one program that tells the promoter to do one particular thing is one cassette. And when the right transcription factors bind, then you get a signal like this, activate me by this signal, and transcription ensues. As you can see immediately, and that's where the picture is still correct, if I take away the first TF1, which says activate, the sentence reads me by this signal, question mark, little to nothing's going to happen in the cell when this factor is missing. While if the factor is missing, for instance, TF3, it reads activate me this signal, the cell might stumble a little bit and you get less expression, but you do get expression. So it's really like a language, including all the redundancy of languages. Let me introduce you to the molecular basis of that. So a more cassette consists of transcription factor binding sites, as already said, in a very strict order, like in a sentence, with some distance constraints in between. This is a more cassette in a promoter that is regulated by circadian rhythm using transcription factor CRAP, which is part of that cassette. This is a more cassette from many promoters are all regulated by circadian rhythm. You see, the structure is exactly the same, but the exact positions of the individual transcription factors become a little bit fuzzy, so they can move a little bit back and forth. We don't invent this, we just read it from the sequence data. And what you see here is nothing else but a translation from what can be seen by everybody just in the sequences. So, formerly we called them TFBS frameworks. And after having seen more than enough imaginary question marks over the heads of people, I told that 2 or 3 years ago, I decided to rename them into more cassettes. Because first of all, it tells you what it is, multiple organized regulatory elements, and it also tells you what it does more than an individual transcription factor binding site. Let me illustrate this. This is the Moore cassette for circadian rhythm. And I had to blind it in part because that is a yet unpublished project with the NIH in Bethesda. So crap is a sure bet, and there are 2 others involved. And with this Moore cassette, we find exactly 24 promoters in the whole rat genome. Pretty selective. 5 of which are actually crap regulated in circadian rhythm, as you can verify. And by using an approach for a random pick of the same size of promoters from all the promoters and the total amount of matches, you can calculate how much you would expect if this is not associated with anything, just random pick. And we find this almost 6-fold overrepresented. So we wouldn't expect less than 1 to find here. Now, CREP is a transcription factor that you can find in about 78% of all promoters. Not in 24, 40,000. So why on earth is this particular? It's particular because of the organization of these 3 transcription factors. Not because there are 3, but the way they're organized. I will illustrate that to you. What I did here is I tweaked that a little bit so that the program is going to find every instance where the same 3 transcription factors occur in the same size window in a promoter. This time, ignoring the organization, distances, strand orientation, order, doesn't matter. Just a bag of 3 transcription factors in a promoter. Here you see what happens. You find 18,000, more than 18,000 matches, because the order is the really definitive part you need. It's not the factors themselves. And this is not overrepresented at all. That's exactly the amount you would expect from a random pick. Here we have a selectivity of 1 in 2,360. Here we have a selectivity of 1 in 3. Or I rather would not call selectivity. Mind my word, there's not written specificity, selectivity. Because we have no idea about the true amount of true positives or false positives, if they exist at all. So we can only say it's selective. We cannot say it's specific. But these Moore cassettes have one feature that is very, very important for the whole strategy. So what we do is we have the whole promoter space in the human, more than 100,000. We have a reduced promoter space from our experiment, whatever we do, like an RNA expression experiment, EQTL study, whatever. And then we go down and we select till we have very small sets, sometimes really 3 promoters, in which we are finally capable of seeing those Moore cassettes. Now, there might be hundreds of additional matches we just lost on the way. The neat thing is we can take this, put it into the computer, and find back every instance in the genome. As I told you, this is not a model, this is what's really there. So we can just read the genomic sequence and say, where do I find this sentence again? And that way it doesn't matter that we have pruned out a lot in the purpose of getting there. And the neat thing is, you find that almost regardless which subset of promoters you initially used, if you were able to find that Moore cassette, by and large, you will find the same from all different subgroups, so it doesn't matter. And that way you can throw away lavishly promoters, because you will always get them back in the second step. how do we determine whether a Moore cassette we found is actually relevant for what we're looking at? You know, promoters are genomic sequences that are invariable, which means the promoter of a gene that is activated in 12 different cell types in your body, but by different regulation, will have all these Moore cassettes at once, and we will find them. But we're only looking at one biological process, so which one is the one that's relevant? So the way we do this is we take our program model inspector, which finds them, and we just run it over the whole set of promoters from the whole genome. From that, by five-finger math, you can basically calculate the expectation for any size of subset of sequences you would have. Then we do the same thing in all the promoters of our desired set, for which we want to know whether it's the right one. And we calculate what we found in reality, and then it's very easy to calculate an overrepresentation between those two. And if that overrepresentation is more than twofold, that is, I find more than twofold promoters in my specific set, then I would expect from a random pick from the genome, I call this associated and relevant, because obviously it goes along with the function. Now you might ask yourself twofold. There might be hundreds of matches that are not relevant. The others? Why? That's the difference between associated and specific. This is a Moore cassette, like the 3 ones I showed you, that is associated. In this case, it was circadian rhythm. It's not specific for circadian rhythm. So it contains basic blocks. Well, the younger of you can remember from their own experience, the older can recall from their children playing with Lego blocks. Very famous. You can do everything with Lego blocks, and there are always these little things. The same here. We have basic building blocks of only 2 transcription factor binding sites that can be used and reused in hundreds of different functions. And as they get more complex, they start to be associated with specific functions. Now let's have a look at our 3 model. If it's extended in one direction by 2 others, it's associated with a completely different function. So that with 3 is associated to more than one function. Or we extend it in the other direction, we get yet another function. Or it's long enough that I only find it inside my functional group, and we do have many examples of that, then it's specific for this function. Most of the time, unless we know, we consider each Moore cassette as incomplete. So it's relevant, it's associated, but not specific. Now we need other means to find the relevant targets of that. We'll come to that. First of all, a few more things about basic concepts of thinking behind all of that approach. So transcriptional regulation is in a way fractal as a fern. You start with nucleotides that consist of atoms. You go to genomic sequences that consist of nucleotides. Transcriptional elements consist of many genomic sequences. More of them combine to Moore cassettes and so on. We get into pathways, we get into pathway networks, and we finally get into cellular networks. But basically, it's all going down, and it's all structured the same way. There's always almost identical subparts that multiply and that form the higher part. What you see here, and this is a very important consequence, as we go higher and higher, the default becomes broader and broader. So there's more and more possibilities what is all possible. Which poses a little problem if you want to find something specific, because the higher you go, the more unspecific it becomes. Here's a workflow. How do we go about finding and evaluating these regulatory elements called Moore cassettes? The very first thing we need is promoters, because they're found in promoters. If I'm hunting for deer and they're in the woods, it's not very good to hunt in the plains. And we look for Moore cassettes, they're in promoters and sometimes enhancers, but promoters are readily available. So whatever list of genes with a common biological function we get, and it doesn't matter whether it's proteomics, transcriptomics, metabolomics, everything that puts genes together in a functional group is amenable. We first use our tool, Gene Promoter, to extract promoters from our database. All of you have access to that. But if not, I don't care where you take your promoters from. You need promoter sequences, period. Take them from wherever you want. And then comes this part that we have to reduce the promoter set because there are far too many to analyze. And I told you that's not dangerous because we'll get back what we threw out erroneously. Then we use the tool called Frameworker, which finds and selects more cassette. And it selects them by the basis that they should be present in a certain threshold of promoters that you define, user define. So if you give it 10 promoters and say, I want that more cassette to be present in 5, I don't care which one, then it will only look for more cassettes that are present in 5 of these 10 promoters. Can be dozens, can be none. So you get an initial set of more cassettes. And then we use Model Inspector, and we go back to the whole set that we pruned and say, okay, so how many more are there? And now we go onto the genome, as I told you, and find everything. And then you can go in and use GeneRanker or JEPS, our pathway system, our enrichment tool, or David, or Ingenuity, or I don't care, whatever is available to find associated pathways or diseases, which is statistics. As you will see, I have a, I would say, differentiated relationship with statistics. So I never trust statistics much farther than I can throw a piano, which is not very far. So I want other things. So I look at additional lines of evidence using other tools, whatever I can get my hands on, pile that up, and finally using something simple like Excel or PowerPoint, or if you're using a Mac numbers in Keynote, compile a final list of relevant genes, Morse, TFs, pathways, and diseases that now are on spot. And as you can imagine, this conundrum is very important for that. So I'm going to explain that in a bit more detail to you. So what are these lines of evidence that we're using? Well, the basics where we get things together for us and for our approach is gene IDs. That's the common platform. Whatever we can pile onto a gene ID is a line of evidence that we can use. Genomics, epigenomics, transcriptomics, proteomics, metabolomics, easy. They're all gene-oriented. Lots of databases and literature are also gene-oriented. Sometimes not directly, but we can figure that out. And a lot of experimental data are also gene-oriented. So we can use this, and then we can integrate these things together. And the integration is usually, for us, in the form of regulatory networks. So, now comes a pretty important part. To all the people who do proteomics or epigenomics or transcriptomics or whatever. All the omics are like doors, like these doors. People usually don't realize it. We go through these doors and come up on the other end, in the room. What you're going to see, if you see anything here, no, you don't see the movie, is that it's basically coming up in the same room. Different perspectives, and each of the doors is one line of evidence. They all point to the same biology from different perspectives. What you see is not identical, but it's a different perspective of something identical called biology. If it's not, something's wrong. It's not willing to go. So for some reason, it probably tries to play the movie in the background, but it doesn't. Okay. Which is not exactly the way it should be. let's try again. Okay. Now it works. So again, we go through all these doors, and in the end, we end up with one biology, multiple lines of evidence which correspond to the different doors we walked through. Now let's have a look at these lines of evidence. What am I talking about? For instance, GWAS SNPs. Some genes do have them or are associated with them, some do not. That's indicated here. We do have differential expression data. We can overlay these, which is already a third line of evidence, the combination, not identical to either of them. We can look at the more enrichment, as I told you, the relevant more cassettes. We can look at pathway enrichments. We can look at pathway overlaps. Now, all of these have one thing in common: arbitrary thresholds. We determine when we call a gene differentially expressed. In reality, it's a continuum. And believe it or not, none of the cells in your whole body has ever heard anything about a p-value. That's a simple fact, but basically that's why they don't behave according to p-values. But we have to use some statistical means to get the search space restricted. So we use statistics. But it's used to pick up the Ariadne thread somewhere and then forget about statistics. But without the statistics, we could never start because I can't analyze the universe at once. So the idea is, as soon as you picked up the thread, reduce those thresholds dramatically. Everything that's spurious will be sorted out by this process. Overlaying that and have a nice pile up of lines of evidence. And they're only going to pile up on relevant genes. So irrespective of any p-values, this is the way to get to it. And to formalize this a little bit better, what we do is like that. So we have our set of genes. We use, like GWAS SNPs, one line of evidence. We pile on top of that another one, like differential expression, and pile on top of that yet another one, and so on, to the amount of evidence lines we do have available. And that depends on the data you have available. That's different from each case. And then you can do that little trick and say, okay, I'll introduce a threshold or a cutoff. And in this case, the threshold was at least 5 lines of evidence have to agree. Some are higher, 6. If available, 7, but at least 5. And that defines a network, because all these genes are connected in 5 different lines of evidence. And this network is a relevant context, because we looked at all the evidences in combination. And they can be accumulated also without hierarchy, which means this guy, for instance, doesn't have any GWAS SNP associated. It's still in there because all the other lines of evidence point towards this is relevant for what we're looking at. So you pull in additional genes, even that were not in your lines of evidence, by looking at the integrated biology, which brings in the genes. And this might have been a fantastic SNP, but in that context not relevant. It doesn't find any further support, so it's out. In that way, the p-value business guided you, but it's no longer important. But it was important. Use this where it belongs, and then stop. Go to other things. There's another problem here. Deduction. Downward is easy. You've got a more cassette. You can easily look at the individual transcription factor binding sites. You can easily look at all the nucleotides behind that. The other way is a bit more complicated because I told you already that a more cassette might be involved in 4 different pathways. So which one is a relevant one? You cannot tell from this data. There's no way. But this more cassette came from a group of genes. And for that group of genes, we can use other additional lines of evidence from the literature, from the databases, from other experiments. And we can overlay that. And where all these lines of evidence cross, that pinpoints the relevant pathways. Could be more than one. So you need the additional lines of evidence to deduce upwards. And that's what we want to do. Okay, enough of that. That was all the theory. Now let me show you a few things that are real data. Study done, of course, with UMISH. Pima Indians, very prone to diabetes and nephropathies. Diabetic nephropathies, which is a chronic condition. And there has been done a wonderful study, found jivas and developed EQTLs, which is SNPs from the jivas that are associated with changes in gene expression. Changes in gene expression, promoters, more cassettes, we're in business. You know, for somebody who only has a hammer, the whole world is a nail. So whatever I'm looking at, I'm trying to find a way to promote us, because then I'm in business. How on earth could an EQTL influence promoters? So that's what we know from the EQTLs. It's associated with change transcription of a number of genes, and actually has to somehow influence the promoter. So there are several ways it could do so. The easiest is there's a common more cassette, and that common more cassette might be even found in an enhancer, and the SNP is in the enhancer deal. The SNP could be something that does an epigenetic change that affects the enhancer, and then we don't need a more cassette in the enhancer, but we use the more cassette to define the mechanism. And then there are other mechanisms that do not involve more cassettes, that do not involve the known enhancers of more cassettes. Unfortunately, biology doesn't always go by one principle. We can look at that. We're content to look at those that do have this. So, they have in common a more cassette. And this more cassette, by definition, defines a regulatory network. As I told you, since it's incomplete, that regulatory network is not going to be all relevant. It contains the relevant parts and some irrelevant parts. But inside the network, I can find associated pathways. And the pathways are known to be correlated already with a phenotype. And in that way, going backwards from I have a bunch of related pathways, I can look at them and say, okay, this one's relevant, this one's not relevant, using additional lines of evidence, clinical data. But as you will see, we don't even have to do this. So that's the principle. Now let's go about it. What we did is we had more than 10,000 EQTL associated genes and transcripts. They were put into 30 modules just to subdivide that as an initial training set. And then we did the standard enrichment analysis in these modules. And we did the detection of more cassettes. And we got more than 30 enriched more sets for that. Explain that in a second. More than 2,000 genes. So it's 20% of the data we can explain that. Unfortunately, it's not all going the same way. But 20% on a genome-wide study is quite a lot. We found a bit more than 180 pathways associated with the standard enrichment analysis by the modules, and less than 100 with the enriched more cassettes. And for the standard enrichment analysis, that's the end of it, because you can't get any more data. Not quite true. You can go into network building, Bayesian networks, whatever you like to do. And then you get networks, but you lose the molecular mechanisms that way. That's the important difference here. We don't lose that, but we get additional information here. We get an idea about pathway gene and transcription factor association, which is a wealth of information. We learn about pathway and more cassette association, getting into more relevant PFPS. And of course, the same way we get pathway and network associations. So that's neat, isn't it? Now about the more sets. More are more or less similar. If you look at the top two here, or the first and the third, then you see they have the same factors. It's just the middle one is shifted in distance. So it's not the same more cassette. They are two different more cassettes, but somehow they are very similar. So, and if you look at this, and that's what I did here, you find a number of them that have a lot in common, and you can do kind of an alignment here. Say, okay, so those are lookalikes, more or less. And then it's a surprise, because you look at some others which don't seem to be lookalikes. But if you look at the matches genome-wide, and you see they're 100% subset of the others. So we threw them into the set. So you can define them by what kind of genes do they find. And if that's always the same, well, you're in business. How does it work? We search for them still individually. But then we combine the results. Why? That way, the selectivity of the individual more cassettes is retained, but the flexibility of the different more cassettes is introduced. If we would put them together and make one big more cassette, you'll get the genome back. Because this is islands that are allowed, and there are vast spaces in between of combinations that are not allowed. It's a similar thing that we did 15 years ago with the matrix families, and it worked very well. So, a little bit more on that. What did we find? Astonishing, in these 30 more cassette sets, we only find 12 major transcription factor binding sites that occupy 78% of all the binding sites, individual binding sites in all these more cassettes. So they're going one direction. It's not random. We have a total of 50 TFBS involved, but many of them are like salt, only peppered in. More than 40 pathway target TFs. If we go for the targets that are associated, they target more than 40 TFs. They're not identical to the 50 up there, but they do include 9 of the 12 major TFs. You see feedback mechanisms coming into play here. And more than 20 target TFs are actually targeted by more than one pathway. It sounds familiar to you, doesn't it? So this is where you see signal integration, all coming up on its own. And if you look at these sets, the 10 sets we have in there, are also not just random. They were all independently developed. But if you look at them, then you can see, and you can read that as follows. 4 transcription factor binding sites that are part of G5, or actually their transcription factors that bind there are targeted by the others. G4, G1, G8. So there's a hierarchy. You can clearly say that the target genes of G5 are probably downstream of the others. Gives you another dimension. It's not just plain, it's hierarchically organized. And this goes on. If you look at these 10 sets, the major sets that contain more than one more cassette, they hail to a number of biological functions. These are the major functions. There are more, but this is also not yet published, so we have to be a bit careful about that. The first function I revealed is cell proliferation, which is associated big with G1. The height of the block for the biological function is the amount of pathways in there. And the thickness of the line is how many of these pathways are actually associated with that more cassette set. And as you see, G2 comes in here, G3 comes in here, and then we have a few more associated down there. The different more cassettes that go for different functional blocks. And there's more, which I didn't show because they're individual pathways that will be too revealing, where they're associated. So we have a clear functional association, and this again is differentiated. It's not random. Now comes a big question. This all is wonderful. This is all consistent in its own right. Has it anything to do with nephropathy? Because at this point, this is a data analysis, and the whole system so far has been absolutely oblivious that we're looking at nephropathy genes. It was a general analysis. So we could find something that's kidney disease related, which would be certainly relevant. Maybe we end up in something that has to do with arthritis or something really outlandish. Let's forget the outlandish and let's forget this. We're interested in kidney disease. What does the world know about this? It's not that it would have been unknown so far. There is a lot of knowledge out there. And the point is that we never used any of this knowledge so far in the analysis allows us now to go in and control it with this knowledge, which of course can be found in the National Library of Medicine. And in this case, more clearly in the medical subject headings, they're all eminence controlled. That is, eminent scientists have put things there. And there are more than 1,400 genes associated with the mesh term diabetic nephropathies, which I hope we all agree is relevant in this respect. And this is associated with more than 200 pathways. Now, that doesn't mean that 200 plus pathways are specific for nephropathy. The same, as I told you, for more cassette goes for genes. They are part of many pathways. So these 200 will contain a set of pathways absolutely unrelated, but called by the same genes. I don't care. I accept this. This is the noise I accept. We have the same noise in our data. The big question is, if we now compare this, our pathways that associated with the more cassettes and the pathways that we call the gold standard, from what all the experts think is important, if our data set would have to do with nephropathies, there should be an overlap. As you can imagine, if there's an overlap of one pathway, I would be hiding away in the corner but never giving a talk on that. So it's about 50% that's the overlap. That's quite good. Taken into account that the blue part has never seen the word nephropathy in the whole analysis. It was just data-driven. Let's go a bit deeper into that, because that is very illustrative. What you see here is a spreadsheet turned into a graphical view. Across are the pathways, and down there are all the genes, individual genes, that actually statistically called that pathway. And what you see immediately is there are a number of pathways that are very, very similar. Pathway database, unfortunately, is very redundant. And across up there, you see that there's a bunch of genes that apparently show up in almost every of these pathways. And for some of these pathways, they are the only reason the pathway is called. So if we look at our scheme of the hierarchy of the more cassettes, then we see that this set of pathways, that's the overlap with the nephropathy genes, is actually called only by majorly 3 of these sets. Nothing random here. Very strictly selected towards specific sets of more cassettes. And all the pathways here, with the green line, merge into 2 pathway modules, which I cannot reveal in detail here, but that's a common function. What I do reveal here is that the function is cell proliferation. They all have to do with cell proliferation. And they all come together. And as I said, some of these pathways are only there because of these 7 genes. It's 7 genes that form the modules. So you see a strict focus of things coming together. And already summarizing part of it. So we had more than 3,000 eQTL transcript pairs virtually distributed all over the genome. It looks about as random as you can think of it. If we go on, it comes down to less than 100 mores, which comes down to less than a dozen functions. And if you look further into that one function, the cell proliferation, and you see that more than half of the pathways are actually called focus on the same modules, the same biological function. So it's a big funnel. And in the end, we get a lot of more information here. We do learn about the pathway network association, as I've shown, more cassette, and the TF associations. Just 12 TFs and manage all of that. And there are 2 take-home messages here. Nothing is random. We start from seemingly random data, but it's all very much ordered. It's not only very much ordered, it is merging onto a focus, not just cell proliferation. There's a bit more than that. But it's really a handful of different biological processes, all of which are known to be important in their properties, where things merge onto. And that way we think that we had contributed a little bit to bridge that cleft between the statistical analysis on EQTLs and the clinical phenotype. Now, it's not Christmas and I'm not Harry Potter, so the point is we can't bridge the gap completely. What we can bridge is the gap to biological function. So we close that gap between the statistics and the molecular biological function using one more cassette at a time, building the bridge for that. So yes, what we achieved is to do this. So we connected GWAS, at least part, by a molecular mechanism represented by more cassettes, with a phenotype, in this case, important pathways for nephropathy, but not that way, unfortunately. As I showed you, it has to go through a little detour through that whole mechanism, but it does work in the end. And I think this is a very nice demonstration what can be done with the data in a purely data-driven way, not biased by knowledge, controlled by knowledge. And that would have been absolutely impossible without the help of the people, well, the main work of the people of the UMISH here. I just contributed to that. They did the main work. Here's the whole group, and Matthias Gretzler, of course, is the head of this group. He enabled the whole thing to be done. And for that reason, I awarded him a special button here, which he agreed to accept. It's called I Love More Cassettes. And with that, I thank you for your attention, and I'm open for questions.
So you've chosen to sort of use a p-value cutoff, and then say to get to the next stage, you have the pathway gene, etc., has to be targeted by 3 or 4 or 5. An alternative is to figure out a way of combining all p-values, because, for example, if you have a very, very strong p-value from one evidence and mediocre but still somewhat significant p-values from the other factors, in my view, that might be more significant than something that's borderline in 5 different pathways. So why did you choose it in the way you did?
Thank you very much for the question. I didn't mention that, but the reason for that is there's a fundamental difference between statistical significance and biological relevance. Lots of the genes that are very relevant here have no p-value at all. They are not significant in any respect we could look at. They are being brought in by the other lines of evidence without p-values because they're important, but they are ubiquitous, so they will never show up. Transcription factors, for instance, that are binding sites that are part of these more cassettes are ubiquitous, like NF-kappa B. You can find them everywhere. What makes them relevant here is the context. And the context doesn't come by p-value. And that's the reason why we don't do this. And as a personal remark, I don't believe in more or less significant. Significant is like pregnant. You are or you're not. I can't believe it's more significant. I know, but dabbling with quantitative levels of p-values, I found that very destructive, and very often pathways that are 100 times more relevant in the end than the top-notch ones have p-values that are really mediocre. And if you go only for the top ones, you will never find them. So I agree, I need that, and I use that to get things started. But as soon as I get it started and I can pile up lines of evidence, I forget about statistical significance in order for finding biological relevance, rather. But the last remark was personal opinion. As I said, I'm not a statistician, so I'm suspicious of that. But I have seen that p-values can be really misleading in that analysis because they rule out important genes. That's what I found in many other cases. Mm-hmm. Yeah, it will work in part. We know that there is a class of enhancers that works the very same way as promoters work, via more cassettes, actually via the same more cassettes that you can find in the corresponding promoters. That's rather easy because we can find them by just searching the genomic sequence. And what in this case would be the best way is to go for the LD blocks and look for additional matches outside promoters for the more cassettes in the LD block, which is most likely an enhancer. Then go to encode other lines of evidence, see that there are enhancer marks there, and then look whether there is a SNP inside one of these binding sites of the enhancer. It's not necessarily a tag SNP because all the SNPs within the LD block are even. They're equal. And so that might be a causative SNP, but not the one that was picked by GWAS. Doesn't matter. You would have a causative SNP. If it's an epigenetic SNP, it's somewhere and you can't tell, of course. But this is one class of enhancers. There are other classes of enhancers that clearly do not work by that mechanism. We have no handle to get to them. Other than that, we could also use the ENCODE data and see, for many, many genes, they have defined enhancer-promoter pairs that correspond. And unfortunately, podocytes, for instance, are not part of that. But you could look for other tissues whether you'll find that. What we actually did is we looked through the ENCODE data with the particular transcription factor binding sites, one by one, that are part of these more cassettes. And we find that about 1/3 of them are found in ENCODE to be bound by the respective factor in other tissues, which tells us that we are picking some biologically important binding sites, but there is nothing that we could look at in the kidney tissue. So the enhancer story is for those enhancers that confer to more cassettes, we can do it. For the others, not this way.
Hey, I was wondering if you find that certain transcription factors very rarely cooperate with other transcription factors and work on their own, whereas other often cooperate with several other transcription factors in a large more cassette. So, like, what's the distribution of numbers of TFs in the more cassette? You know, I'm just wondering about certain properties of these.
We have looked at that in great detail because we have our module library, which has more than 800 experimentally verified interactions of transcription factors. The unfortunate thing is they are all tissue specific. So two transcription factors interacting in the same promoter in one tissue may be absolutely meaningless in another tissue. There are some groups of transcription factors that very often interact in biological functions, like, for instance, STAT, NF-kappa B, IRFs. You find them over and over again in all immune functions, not only in one cell, B cells, and T cells. There are other factors like CRAP and NF-kappa B that tend to interact. And this is interesting because you find that quite often when there's epigenetic regulation involved, because they both call CP300 an epigenetic regulator. So there are some data on that. We don't have a general statistics on that due to the diverse tissue specificity, which we cannot analyze. But we do take into account known interactions, and they're all listed in MudBase. So you can find there which interactions are known for each and every transcription factor. And there are more frequent ones and there are more rare ones, but I can't tell you percentage or something like that. But individually, you can look it up.
Do you think your approach to identifying the more cassettes is biased towards finding smaller or larger ones?
Actually not, because what we do, we can also go for more cassettes of 2. The program does allow for that, but you're drowning in them because there's so many basic building blocks for that. But starting from 3, and sometimes you have a more cassette of 3 and you have one of 4, which is an extension of the 3, and they have quite distinct matching patterns. We are probably biasing towards the shorter ones, 3, 4, because the combinatorial tasking of the program with larger ones becomes really, really hard. So if you have too many promoters, then you will tend to find the shorter ones. That's why I said we always consider them incomplete unless we know they are complete, because basically we'll find them incomplete. So yes, there is a technical bias towards shorter ones. Shorter ones means we still have fingerprint more cassettes that are once in the genome. There's one promoter in the genome that has them, and if you analyze the mouse genome, you have one match in the mouse genome, which is the same promoter. So the selectivity specificity is not affected by that, and sometimes they have only 5 or 6 elements. But it's rare to find 8 or 10 elements. That's clear. So our working horses are usually 3 to 5.
Yeah, I had a similar question regarding evolutionary conservation. How you define can you define it better with integrating more the species, data of more species to define these more cassettes?
Excellent question. We do both vertical and horizontal analysis for this. And if you do a vertical analysis, human, mouse, rat, whatever, the more cassette you define is not associated with one function, unfortunately. It's associated with the combination of more cassettes that are conserved for several functions, because you can't distinguish that. So they're rolled into one. If you combine, and actually they're conserved throughout the mammals, and sometimes down to Fugu. Not farther, but down to Fugu in part. And if you do the horizontal analysis, they're usually associated with the one function you're looking at. And if you combine that, then you see they're different, but then you can see the functional one in part in the vertical one, or not at all. Because we have a significant amount of species-specific regulation that is visible in the more cassettes. While you do have the same gene has a different more cassette in mouse than in human, because the regulation is species-specific. In those where it's conserved, which is still the majority, you can see that. And yes, you get different aspects from the phylogenetic conservation. That's cool.
For the nephrologists in the room, this, for example, worked for podocyte-specific transcription factor binding modules who allow to, from a seed set of molecules, predict future podocyte-specific genes. Several of them turned out to be even associated with FSGS. So there is evidence that combining both horizontal and vertical domain can pull meaningful candidate gene sets together. Okay, if no further questions, thanks a lot. Thanks a lot for the attendance.
Thank you.